All right. Hello everyone. Welcome to our today's Zoom seminar. It's really my pleasure to welcome our today's speaker, Dr. Rachel Gottschalk from Pittsburgh. Rachel currently is assistant professor in Department of Immunology in University of Pittsburgh. Rachel completed her undergrad study in Emory University and then continued with PhD training in Cornell in New York. And then she moved to Maryland to NIH, did her postdoc training with Ron Germain in immunology and system biology. From 2018, Rachel started her independent research program as assistant professor, as I just mentioned, in University of Pittsburgh. Rachel's lab is really trying to understand how excellular cues, how they link to a gene expression, and particular innate immune signal, how they integrate in different scenario between the health and condition versus inflammation. Her lab used quantitative experimental and computational approaches to address these questions with main project including several aspects. For instance, like computational modeling of signaling to transcription in macrophage, which I think we're going to hear about today. And also her lab trying to um, interrogate long-specific macrophage signaling, and then last but not least, to dissect the molecular determinants of monocytes and macrophage inflammatory functions. Her lab using both data-driven and mechanistic modeling approaches to integrate uh, transcription factor activities, global phosphorylation, and then transcriptomic data to explore signaling mechanisms which shape stimuli specific macrophage function. And then by doing these, her lab will yield insight into dysregulation of signaling and inflammation while informing therapeutic strategies. Um, Rachel did a really quite productive, both the PhD and then postdoc training. And then with that, she received numerous awards, which I'm not going to list all of them. I just want to emphasize, like, she became a member of NIH Maximizing Investigator Research Award from 2025 to 2029. It's really exciting to have Rachel with us tonight, and then we're gonna hear about the signaling in the macrophage function. So thank you so much, Rachel. We are very much looking forward to your talk.
Sorry, I think I just pressed something. Can you still see my screen?
I can, yes.
Okay, great. Thank you so much for inviting me. It's really a pleasure to get to present to um, people, I guess maybe all over, who would not normally get the chance to interact with. So I look forward to, answering questions at the end. And then if anyone is interested in chatting more, receiving emails after the talk. Um, And thank you for the introduction. So I think it really gave you a good sense of like the types of things that my lab is interested in. So today I will give quite a little bit of a background, even though I know that most of the audience is fairly familiar with immunology. So as most people know, macrophages are present in pretty much every tissue of the body. And so in recent years, people have become more aware that these cells have very important functions in homeostasis, including tissue-specific functions. So, for example, in the spleen, so these cells participate in kind of clearance of red blood cells and iron homeostasis. In the lung, which we'll talk about later, in addition to taking up particulates and being anti-inflammatory, these cells are really important for surfactant homeostasis. And so it's becoming really well appreciated that these cells are not just innate immune cells, but also have these really important tissue-specific functions. Obviously, what we first learned about in Janeway or a basic immunology textbook is the important role of macrophages in inflammatory response regulation. So These cells are considered really the first line of defense against pathogens. They sense microbial products that results in production of cytokines and chemokines and really initiation of an inflammatory response. And so I really like this illustration here because it shows how both the tissue resident cells and then the incoming monocyte-derived macrophages really are important for all the phases of the immune response. They initiate inflammation, they kind of control how the magnitude of inflammation, and then as the complex cues in the tissue change, they really are playing a pivotal role in sort of shifting to an anti-inflammatory response, really to resolve inflammation and to return the tissue to homeostasis. And as I won't talk a lot about disease today, but when this process is dysregulated, it results in a lot of macrophage-associated inflammatory pathology. So what I like about this image is it really kind of, with these arrow sizes, it really highlights how different parts of this process, whether it's kind of the scaling of the pro-inflammatory response or the transition towards the resolution response, when this isn't appropriately controlled, it can lead to either too much inflammation or prolonged inflammation that can drive disease. And so how do the cells know what to do? So this is really kind of getting into what we're really interested in is all of these decisions that the cells make are resulting from their integration of tissue cues, right? So in homeostasis, the cells are receiving cues from the tissue that really is driving their tissue-specific homeostatic function. And then cells that are present in an inflammatory environment, they're receiving additional cues that are telling them about what's going on. Are we at the initiation of infection? Or is the pathogen alive? Is the pathogen dead? Or is there tissue damage? And all these cues are being integrated to really drive the appropriate gene expression and functional programs. And really, quite a lot is known about this. So, for example, with the tissue-specific macrophage functions, there's been a lot of really beautiful work done in a variety of tissues, really building on this paradigm where you have tissue-specific cytokines and other factors that drive the activity of sets of tissue-specific transcription factors. And these transcription factors are really critical in what we call programming of the tissue macrophage. And so this, how the tissue programs the macrophage to achieve its transcriptomic and functional state is something that's been of a lot of interest the last maybe 15 years or so. And so a lot of work has been done in this area. And then likewise, a lot of work has been done in the area of regulation of inflammatory responses. And so this is, again, showing you kind of with this transcription factor focus that quite a lot is known about the stimuli, the signaling pathways, and the transcription factors that drive pro-inflammatory and anti-inflammatory responses. I would suggest that less is known about the intersection of these processes, this tissue-specific programming and the inflammatory response regulation, and this is one area that we're particularly interested in. And so my lab generally, the common theme in the group is kind of thinking about using quantitative, experimental, and computational approaches to really understand this decision-making process. And so we have a lot of different projects, different stimuli, using different types of computational approaches, and really focused on this signal integration process where the macrophage just takes all the complex inputs and decides what's the appropriate program. And today I'm going to really focus on two particular research areas in the lab. One is related to transcription factor interactions that underlie macrophage function in the lung and alveolar macrophages. And the other one is thinking more about more the systems biology side of the lab, where we're thinking about how do we link transcription factor activation features to specific genes. And at the end, just a few slides, I'll really talk about how kind of our future goal is to sort of link these two research programs together. So my foray into alveolar macrophages really started when I came to Pitt. I had been working in bone marrow-derived macrophages and really wanted to start thinking about tissue-specific macrophage signaling, and was very interested in tissue-specific cytokine signaling. And so what you're looking at here is data that sort of motivated our first paper related to alveolar macrophages, where we're thinking about how, okay, if different if different cytokines are active in each tissue, how are these kind of constant or steady state cytokine signals regulated? And so what you're looking at here on the left is looking at expression of these SOX family members. These are inhibitors of cytokine signaling, and you're looking at their expression across tissue macrophages. And we noted that CISH was really specifically expressed in lung macrophages. And because this is published, I'm really going to just show 2 slides because it kind of motivates the ongoing work in the lab. But we basically were interested in how the role of CISH in tissue-specific macrophage function in the lung. And we noticed in the CISH knockout mice that with age, you sort of start to acquire these kind of enlarged alveolar macrophages that really kind of take on this foamy macrophage phenotype seen when you have surfactant dysregulation in other models and in human disease. And so, and we did notice this kind of increased granularity and enlarged cells, sort of again, is consistent with this foamy macrophage phenotype. And we did also see some evidence of surfactant dysregulation in these mice. And we performed RNA-seq. We sorted out alveolar macrophages from wild type and CISH knockout mice and performed RNA-seq analysis. And again, there's lots more information in the paper, but really this kind of as a jumping off point for the unpublished data, we found that in the differentially expressed genes in the CISH knockout alveolar macrophages, there was a really strong enrichment in motifs associated with GATA transcription factors. And so GATA transcription factors are obviously really important in a lot of other signaling systems, including hematopoiesis and other types of immune cells, but typically haven't been studied as much in mature macrophage populations outside of GATA6 in the peritoneal cavity. And so we thought this was really interesting, and we kind of interrogated all the different potential GATA transcription factors and noted that GATA2 was specifically expressed in lung macrophages. And so at the RNA level, it's still relatively lowly expressed, which was something that was sort of a barrier to progress for a while. But actually, a recent paper came out that shows that it's tightly controlled at the post-transcriptional level. And actually, we were able to detect GATA2 protein in alveolar macrophages. This is just showing you cyclic F positive alveolar macrophages in the lung and looking at phospho-GATA2 serine 401. And so we decided to go ahead and take the dive and figure out, okay, is GATA2 playing a role in the tissue-specific function of alveolar macrophages? And we generated myeloid-specific conditional knockout mice. I'm kind of not going to show a lot of negative data. It turns out that these mice really have no observable pathology, and most of the homeostatic functions of the alveolar macrophages are intact in the GATA2 knockout mice. But we did get some interesting results in a type 2 inflammation model. And the reason we went this direction is because obviously GATA3 has been described as being very important in type 2 immune function. And so the model is basically we challenge with IL-33 intranasal, and 24 hours later, we quantify inflammation and also alveolar macrophage gene expression. And this is just showing you that you do get a robust type 2 response in the alveolar macrophages. I'm not showing it here, but this is not direct sensing of IL-33. Macrophages in the lung don't really express the receptor for IL-33 in a steady state, but instead is due to sort of indirect effects, likely ILC2 mediated secretion of IL-13 and IL-4 downstream of IL-33. But you get this really nice, robust type 2 immune response and nice changes in gene expression. Again, this is the sorted alveolar macrophages. And we were able to see a lot of different expressed genes in the GATA2 deficient macrophages. And so we took all of the DEGs from the GATA2 versus wild type, and we performed a pathway enrichment and found that there was an enrichment in pathways associated with metabolism. And so here, this is just showing you some of the top pathways that came up, and then here is just a heat map of some of the metabolic pathways. And I'll kind of point out again, consistent with what I told you earlier, we're not seeing a ton of differential expression in the steady state, but in this type 2 inflammatory response, we're seeing significant differences in genes in these metabolic pathways. So we went on with our collaborators, Ali Cohen, does a lot of immunometabolism work, and with their help performed seahorse metabolic flux analysis, and did find that GATA2 deficiency does dysregulate the metabolic response in these alveolar macrophages. So if you focus on the top graphs, you can see in the steady state, there's actually sort of a modest but statistically insignificant across experiments increase in the metabolic activity of the alveolar macrophages. However, at 24 hours after IL-33 exposure, so when you isolate cells from this inflammatory environment, we're actually seeing this decrease in the metabolic activity of the macrophages, sort of suggesting that when you put these macrophages through this inflammatory stimuli, in this context, they're not able to, you could think of it as, I've heard her say like metabolic collapse, but basically the idea that they're not able to kind of shift their profile to kind of accommodate this inflammatory setting, suggesting that GATA2 is important, sort of important for the metabolic plasticity of these cells. And so we haven't yet, this is ongoing work, we haven't yet really gotten into the mechanism of what is going on with the metabolism in the absence of GATA2. But because GATA2 is a transcription factor and actually in other cellular contexts has been shown to interact with known regulators of metabolism, we wanted to start by thinking about what other transcription factors GATA2 might be interacting with. And so this is a figure that I really like, that shows kind of the role of GATA2 in adipocytes. And you'll note here, any alveolar macrophage people in the audience will note that a lot of these transcription factors that GATA2 is known to interact with are actually really critical for lipid metabolism in alveolar macrophages. And so we took the differentially expressed genes and we just asked, using this CHEA3 transcription factor target enrichment, in an unbiased way, we asked what other transcription factors are sharing targets with these differentially expressed genes. And kind of consistent with this figure here, we noted that PPAR gamma came up, PU1 came up. And so suggesting that one reason we might not be seeing a major impact of GATA2 knockout in the steady state, but seeing an impact in inflammation, is that it may be really largely playing a role in regulation of other key alveolar macrophage transcription factors. And I want to note also, in terms of our differentially expressed genes, that CPP alpha is dynamically regulated during inflammation, and this seems to be dysregulated in the context of the GATA2 knockout. And so we're still pursuing this, but the idea is that GATA2 might be really critical in kind of helping shape the activity of other transcription factors during inflammation to sort of allow for that metabolic plasticity of the macrophage. And so this is sort of transitioning on to like a very closely related project where we're really thinking a lot about how transcription factor interactions support the plasticity of tissue-resident macrophages, specifically alveolar macrophages. As I mentioned earlier, I think one of the really key questions in the field is, while we understand the transcriptional regulators of tissue-specific immune function, and we understand regulators of inflammatory function, it's not really well understood how these programs intersect and how a macrophage can sort of maintain its tissue identity while having this plasticity to promote and then resolve inflammation. And so really this question of this project, led by student Sonia in the lab, is how this kind of transcription factor interactions can support the plasticity of macrophages. And so she leveraged a recent data set, a single cell data set published by Bill Jansen's group, where they did this really nice time course where they treated with LPS and harvested cells at different time points, performed single cell RNA-seq. And this data set was really helpful for us as we leverage it to use some different gene regulatory network approaches. And so the first thing we wanted to do is really identify gene topics that were associated with homeostasis or inflammation. So we perform topic modeling. And what you're thinking of here is if you think of like the genes as, think of this as genes that are really sort of defining the different cell states that are present in the single cell RNA-seq data. And Sonia performed kind of optimization of the number of topics and came up with 7 topics, one of which is not shown here because there's just very few cells. And so what you can see here is that this topic 7, which I'll refer to as the kind of homeostasis topic, is really high in this group of cells that's present in homeostasis and at the later time points. Whereas topic 4 is really associated with peak inflammation. And so what we're really trying to do here in these next few slides is identify the transcription factors that help to drive the homeostatic topic or gene program versus the inflammatory program. And so this is just showing you the type of genes pathways that are enriched, kind of consistent with what I mentioned earlier. A major function of homeostatic function of alveolar macrophages is related to the kind of lipid processing, and PPAR gamma is a major driver of this. So this PPAR gamma signaling is consistent with this kind of homeostatic function. And then topic 4 is really enriched for a lot of different inflammatory pathways. So I'm not going to go into detail here, but we leveraged a couple different novel, not made by our lab, but kind of recently published or pre-printed approaches to really try to understand the transcription factors driving these different programs. One of which is Chrome-BPNet, which leverages attack-seq data. We used alveolar macrophage attack-seq data from both baseline and LPS-treated alveolar macrophages. And this approach really allows you to kind of identify de novo motifs and then the associated transcription factors that are really important for the open chromatin in these different cell states. And so using this approach, we could sort of identify a lot of the known regulators of alveolar macrophages, but then and gain additional insight. And so what we expected to find was that we would have certain transcription factors driving the homeostatic program and other transcription factors driving the inflammatory program, and that there would be some sort of interactions between the two. But instead, we sort of started seeing a different picture. So what you're looking at here is really showing you using those contribution scores from the Chrome-BPNet, which leverages the footprinting. You can see that sort of some of the expected players, like IRF and NF-kappa B, are really strongly associated with topic 4, the inflammatory topic. And other players, as expected, like PPAR gamma, you see GATAs, are really strongly associated with the homeostatic topic, so topic 7. But we were somewhat surprised to see that some of these transcription factors, CPB, like CPB beta and PU1, which we think of as being really critical for alveolar macrophage homeostasis, they're sort of off the axis and really equally sort of associated with enriched in both inflammation and homeostatic topics. And so we sort of have built a new model now, and with a lot of results where I'm not showing you due to time, we're using both this footprinting approach, but also Cell Oracle to build gene regulatory networks, where now we have a model where these really key identity-forming transcription factors act really by interacting with both homeostatic transcription factors and inflammatory transcription factors in a very dynamic manner. So what we're seeing is that at the different time points of homeostasis to peak inflammation and resolution, you sort of see the strength of these interactions really shift over time. And even more interesting, we're seeing evidence that some of these identity-forming transcription factors really act by restraining the inflammatory transcription factors. And so it's sort of, I would argue that these identity-forming transcription factors both enable some degree of plasticity, but also restrain plasticity of the tissue macrophages. And so obviously I'm not going into all the details. You could probably imagine these networks get pretty complicated pretty quick. But if anyone's interested, we're actually preparing a manuscript on this project, and I'd be happy to chat with people. And so really what we're putting forward is this model where you have these kind of inflammation TFs that are very much consistent with what we know in the literature. Then you have some of these homeostatic TFs that, such as PPAR gamma. But then a lot of these identity forming TFs really kind of work by kind of allowing the cell to sort of pivot between these two programs. So one thing that I'm not going to talk about much today is we actually have a couple projects in the lab thinking about how external stimuli, so systemic stimuli, so microbiota or diet, how systemic factors shape the lung environment to in turn alter the macrophage state. And this is obviously sounds like a large kind of it's an immunology body-wide problem, but we're really leveraging it to think about cell state and how the altered cell state of the macrophage will change their responsiveness to stimuli. And so I'm going to kind of pivot now to sort of our more systems biology projects in the lab, but then kind of come back at the end to this idea of cell state and how the two projects really kind of eventually will be united. So thus far, in thinking about how macrophages are interpreting signals in the microenvironment, we've really been focused on the inputs leading to transcription factor activity. But when we think about gene expression programs, an important aspect of that is really you have these really diverse gene expression responses. And so another thing we're really interested in is thinking about how these signaling dynamics are decoded at the gene level. So thinking about how you could have a bunch of different genes that are targets of this transcription factor, but they all might interpret the signal differently across different cell states, for example, depending on things like open chromatin, the prevalence of some sort of competitive factor, or the presence of necessary cooperating pathways. And so this is really a fundamental problem. We're addressing it in the context of bone marrow-derived macrophages, but this is just a really fundamental signal integration problem of how signals are interpreted at the gene level. And so the model system that I'll be talking about today is really in cytokines. We're also doing some work in toll-like receptors. But really thinking cytokines are appealing because it's kind of this thing that all immunologists understand of this problem of how is it that many cytokines activate the same transcription factors, yet get these really specific and diverse gene outcomes, and how the same transcription factor could be active in many different contexts. And again, you get these different patterns of gene expression. And so it's a really fun problem to tackle. It's a long-standing problem in the field, but it's a fun one to tackle because though it's an old question, there's always kind of a new way to tackle it. So this is a paper that came out last year. My first PhD student, Neha Chimalavagu, who graduated over a year ago now, really undertook this effort of thinking about how we can use modeling to start to understand this problem. So what we published is this, it's a mechanistic to machine learning workflow where you have a mechanistic model of JAK-STAT signaling. It produces these STAT phosphorylation trajectories, and these trajectories serve as an input for a machine learning model that predicts global gene expression, so from RNA-seq data. And the reason that this workflow is appealing, I'll kind of get into more detail, is that it allows you to actually make perturbations on the signaling level, and then see test all the way through or predict all the way through what would happen both to signaling and gene expression. And so the model system that we were using is IL-6 and IL-10. This is a popular model system for this for the reason that both of these cytokines activate STAT1 and STAT3, yet typically are thought of as having opposing immune functions. And so we generated time course data of STAT immunofluorescence, phosphostat immunofluorescence, and RNA-seq. And then we use our workflow to connect the two. And so this is just to give you an example of these are not, these trajectories are not based on live cell data, but rather sort of fixed time point data. So for each time point, we have sort of, we do have the single cell variation, but we're not able to connect the individual cells across time. So the modeling approach we're using is BioNetGen, which is developed by our collaborator James Fader. And just is just a simple idea, kind of explanation of how it works. And it's really a nice entry point to mechanistic modeling for those who are not trained computationally, like myself, because you really just have to learn to think like a modeler and the kind of structure of the rules. But it's very intuitive in that if, for example, if you know that receptor 1 binds receptor 2, this reforms a receptor complex, and it's really as simple as this is one reaction rule, two proteins, and then you have four model parameters being the concentrations of the receptors, the association rate, and the dissociation rate. And this is basically just the simplest example of like a rule-based model. So they have made a model of JAK-STAT signaling with IL-6 and IL-10, the appropriate JAKs and STAT proteins. Obviously, there's some simplifications in the model, but really this was enough to reproduce the experimental data. So this is just showing you experimental data are the dots, and the simulated trajectories are the curves. And again, the main point I'm trying to make here, even though we go into a lot more detail in the paper about the mechanistic modeling process and the parameters and what we can learn, really what I'm trying to show here is that these trajectories then serve as the input for the machine learning model. And again, um, you could say, okay, if you have enough, you can make simulated trajectories, you can do gene expression. What's the point of this very labor-intensive mechanistic modeling process? And really, it's because this sort of approach allows you to ask really quantitative questions about the system. So how do cells tune signaling networks? So, for example, if you think about like a naive cell versus a memory cell, or a cell that is residing in tissue A versus tissue B, they all can sense IL-6, for example, but they might sense it in a different way. And so, how does a cell tune a signaling network? How can we assess the influence of variation? So does a twofold difference in JAK1 expression matter? These are the kind of questions that you can ask with a mechanistic model. And then in terms of the test case in the paper is how does a change in signaling impact gene expression? And so as our first test case for this workflow, we decided to focus on JAK inhibition. As most people know, there's many different JAK inhibitors on the market for a variety of inflammatory diseases. And while they've been really transformational, there's a lot of open questions. Which patients are likely to respond? Which JAK inhibitor to use? How to predict off-target effects? And so while this is really just a proof of principle study, we do have ambitions to really start to tackle some of these questions with our future approaches. And so we asked, can we use our workflow to predict the effects of a JAK inhibitor? So what we did is we perturbed different parameters in the mechanistic model to basically simulate changes in JAK2 activity. And so then with this workflow, we can say when you make these perturbations, what happens to signaling and what happens to gene expression? So at the signaling level, this is just really showing you compared to the red, which would be sort of your unperturbed trajectory, what happens with going increasing perturbations up to just threefold of JAK2 activity. And you can see with these different parameters by perturbing the JAK expression or the JAK to STAT activation rate, we start to get these sort of modest effects on STAT3 phosphorylation and these pretty drastic effects on STAT1 phosphorylation. And then we were able to validate this prediction using the frijotinib, the JAK2 specific inhibitor frijotinib. So showing you again that as predicted, we were able to kind of get this modest shift in the STAT3 response while really having a major impact on STAT1 activity. So then with these trajectories, we can make predictions about gene expression. This is always the slide that I feel like I tend to lose people on, so I'm going to try my best to make this really clear. So the idea of the workflow, you train your machine learning model on unperturbed. So basically you say, hey, this is what the trajectories look like with IL-10 and IL-6, different doses. And this is what the gene expression looks like. And you train the model to relate the trajectories to the gene expression. Then the testing of the model is you say, okay, here are trajectories that have been inhibited by JAK2, these gray trajectories. And then you say, can you still predict gene expression? If the model still predicts gene expression very well, so this is the prediction error between the training and testing, this is telling you that the model doesn't care about this JAK2 perturbation. This is a JAK2 independent gene expression effect. But if you suddenly have a large error when you feed in these JAK2 inhibition trajectories, this large error is giving you the indication that these genes are highly sensitive to JAK2 variation. And so using this approach, we came up with sort of the genes that would be the most sensitive or insensitive to JAK2 inhibitor. And then we were able to validate that using RNA-seq, IL-6 stimulation with and without fedratinib. And so we were able to show that our predictions could be validated, where genes that were predicted to be JAK2 independent had had a smaller fold change compared to genes that were predicted to be JAK dependent. And so this is a subset of genes from immune response pathways, but this was also true at the genome transcriptomic wide level. So this was, I think, an important example of how this workflow could be used to actually predict gene level responses to signaling perturbation. And to my knowledge, I think it was the first time that people have been able to, again, make these gene level predictions to kinase inhibition. So as a summary here, using this workflow, you can make perturbations to a mechanistic model. You can predict the effects on stat trajectories, and then predict global gene-specific effects of JAK2 inhibition. And this is all published in the Cell Systems paper last year. So what's going on now? I was super proud of this work. Honestly, it's probably my favorite paper that I've ever published. But there's obviously still a lot of knowledge gaps or things that we did not achieve. So if we want to start asking these types of questions about making meaningful predictions about the specific effects of JAK inhibitors, we need a lot more mechanistic understanding of these predictions. So for example, while we were able to predict some genes would be JAK2 independent and some genes would be JAK2 dependent, I would not be able to tell you really anything about why. Why are some genes more sensitive to JAK2 than others? And so this is where it sort of ties back to this idea of gene-specific decoding of signals. And this is something that I'm just really interested in at a fundamental level, is really being able to understand what are the mechanisms that support this sort of gene level response to cytokines. And so just kind of stimulate thought. So as I showed you earlier, when you give the JAK2 inhibitor, you have a much larger impact on STAT1 than STAT3. And you'll note here that while we sort of focused on the kind of absolute value of fold change, there's actually a lot of complex stuff going on with this inhibitor treatment. So when you give the cell the JAK inhibitor, you have some genes that go down. This is intuitive, okay? So you think, okay, you're inhibiting a kinase, you're inhibiting STAT activity, gene expression should go down. So you could say, okay, these are genes that are dependent on STAT1 for their activity. But then you have other genes that actually go up. So genes that are increased with JAK2 inhibition. And so this is where you start to get these interesting possibilities of, okay, maybe these cells that are these genes are indicating some sort of competition because now they're up with decreased activity of STAT1. So maybe there's some sort of competition between STAT1 and STAT3. So these are just some examples of how you have these really gene-specific responses that we'd like to be able to understand. And so ongoing work is sort of like version two of this workflow is now leveraging a sequence to function model. So our colleague at Pitt in this computational biology department, Maria Chiquina, has a lot of experience with sequence to function models. And the idea here is now we're not just feeding in the stat trajectories, but we're also using promoter sequence. And now we're not just predicting gene expression, but we're also predicting stat binding by cut and run. So we're working towards a much more mechanistic understanding of why some genes are responding differently than others. And the ultimate goal is to really identify promoter sequence features that are predictive of stat behaviors. And so the hope is that if you identify these promoter sequence features that kind of predict response type A or response type B of the genes, this could be related to specific cooperating factors, right? So we can use motif analysis and identify factors associated with, inhibition or activation of these target genes. And so that sort of relates to kind of tying it all together in the last two slides is how once we understand this gene specific decoding of signals, we have a lot more insight into altered cell state. So for example, and this is kind of cell state A versus cell state B. Suddenly, if you have different levels of these chromatin accessibility or inhibitory factors or cooperating pathways, you could give the same stimuli to do two different macrophages, for example, but get a completely different gene expression response. And so this is again showing just kind of how you have an altered signaling state that would impact cytokine-induced gene expression. And so this sort of, I think on the long-term vision of the lab, I'd like to eventually see a future where these two sort of parts of the lab are kind of tied together, where we can start to think about how altered cell state as a result of these sort of environmental stimuli or tissue-specific stimuli would result in distinct types of responses to inflammatory stimuli, and how this would sort of control a cell's ability to appropriately regulate an inflammatory response. And then again, thinking about how altered cell state and this kind of signal encoding and decoding would impact susceptibility to disease. And so with that, I'd like to spend a few minutes acknowledging people. So previous lab members, Carson led the SISH work, which is published, and Neha led the cell systems machine learning workflow that's published. They're both have moved on to exciting new things. Sonia led the work with the gene regulatory networks, and Morgan and Chadarupa led the work with GATA2. Both of those papers should be hopefully out soon. And then just to leave you with kind of these are all a lot of young people in the lab, a lot of students in their third year or new postdocs. And so hopefully we'll have a lot of new exciting things to show you if you invite me for future seminars. And with that, I would love to take questions.